Welcome everyone to today's webinar. My name is Marie Latulippe and I am the Director of Science Programs at the Institute for the Advancement of Food and Nutrition Sciences, or IAFNS. And this is the fifth in our 2021 webinar series in which we're continuing to learn about diet and the impact on the gut microbiome and how that ultimately impacts health. So for those of you that may not be familiar with IAFNS, we always like to reiterate that we are a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health. And we do that by bringing together the intellectual contributions of scientists from various sectors, industry, government, and academia. And together they determine what the issues of common interests are and develop research projects around those common interests. And our operating philosophy is that including those diverse perspectives in discussions is really fundamental to developing credible science and science that benefits the entire food and beverage ecosystem. And another thing that we would like to make the scientific community aware of is a new opportunity that IAFNS has made available. And this is in our spirit of openness and focus on the inclusion of those diverse perspectives. So we've launched an idea portal, and this is available on our website. And this portal is really an open invitation for proposal submissions on topics of relevance to IAFNS, which means that that also is aligned with a public benefit. So this portal is perpetually available and open, and we invite you to visit the link at the bottom of the slide here and submit a concept. Before we start, just a quick note on logistics. So you probably see that control panel on your desktop. Go ahead and submit questions at any time over the course of the webinar today, and we will take those to the end and engage in a dialogue with our presenter at that time. And with that, we are very pleased to welcome today Dr. Vanessa Leone. Dr. Leone is an assistant professor of animal biologics and metabolism at the University of Wisconsin. She completed her PhD in animal sciences and nutrition at UW-Madison. And then as a postdoctoral fellow at the University of Chicago, Dr. Leone examined how the trillions of microbes that reside in the intestine impact complex metabolic diseases such as obesity, diabetes, and non-alcoholic fatty liver disease. And currently, Dr. Leone holds an NIDDK K01 Mentored Research Scientist Career Development Award and focuses on determining the mechanism of how diet-induced gut microbes shape and maintain host circadian clock function in the context of gut mucosal health. And then in that, she has a specific emphasis on antimicrobial peptides. So the overall goals of Dr. Leone's research are to identify novel microbiome-derived strategies that can be used to improve the metabolic health of humans, as well as companion animals, and also to promote agricultural animal immune health and growth and production outcomes. So with that, we are very pleased to turn the platform over to you, Dr. Leone.
Thank you so much for that kind introduction, and it's really wonderful to be here with everyone today. Yes, as was just mentioned, today I'm going to be talking to you about how I think gut microbes are contributing to aligning our circadian networks and what I think is really the metronome of life. And so if you're ever bored, I've written actually a review with some talented graduate students that I co-mentored while I was at UChicago, where we kind of build this idea around a metronome and an orchestration. So to give you a little bit about myself, I, as was mentioned, did my PhD here at UW-Madison, and I went away for a little bit, just a little bit further south to University of Chicago, and then I had the opportunity to come back here to UW-Madison. And so if you've ever been to this campus, it's very beautiful and very collaborative, and it's wonderful to be back. And I'm part of a new effort on campus called the Meat Science and Animal Biologics Discovery Program, and I'll introduce that a little bit to you in the next slide. But I sit in this brand new building on campus that was a $56 million investment from the state and stakeholders to advance research really in this really important area. So my lab has two goals. Some would say they don't overlap, but I argue that they do. So what I'll be talking about with you today is this idea of gut microbiome and metabolic health. So really trying to understand how the gut microbiome contributes to either leanness or obesity, and identifying the qualities that define a lean or eubiotic microbiota versus an obese or dysbiotic microbiota in the context of metabolic health. Now, the other facet of my lab is this whole animal biologics effort, and it's really what we've decided is a way to develop co-products from discards at the time of animal production that can be used to promote health and wellness amongst both animals and humans. And so some of you may be familiar with heparin, which is what we consider to be a biologic. And so this is actually derived from porcine mucosa. And actually that is probably about 90 to 95% of the heparin that's in circulation across the world today is actually from porcine mucosa. So we think that there's opportunities to harness these, to promote health and identify novel molecules like that, including those derived from the microbiota. But today I'm really going to focus on how I think the gut microbiota is contributing to health and wellness. So an overview of my talk is to give you a little bit of a background, talk to you a little bit about the role of diet and microbes in metabolic function, which it sounds like you've been hearing a lot about. And then I'll go over some of the initial findings that connect circadian rhythmicity between host and microbes, and really talking about how did we get here. Go over some of my more advanced findings and go over dietary intake on host and microbes, small intestinal microbiota oscillations, and really highlight how I've been using in vitro and in vivo studies to identify and gain some mechanistic insight. And then hopefully I'll give you some key takeaways at the end that you agree with and we can discuss. So this is not new to anybody attending this webinar today. So the prevalence of what we call new age disorders has really increased over the past half century. So obesity and type 2 diabetes have been on this progressive march. And here we have maps of the United States highlighting this increase in incidence of self-reported obesity as well as type 2 diabetes across the United States. So as of 2010, over roughly 26% of the US population was considered obese. And I always say that it's really bad when the CDC decides to recolor these maps, and this is probably even a slightly outdated statistic at this point. But now, you can see that in certain states across the US, there's greater than 35% obesity. And this is a major problem because we don't really have many interventions to alleviate obesity or even prevent it for that matter, aside from some major lifestyle changes and then a very drastic intervention of bariatric surgery, which also has some very interesting microbiota implications surrounding it. And we know that the development of obesity and metabolic disease is really multifactorial and complex. So of course, there's a genetic component to many new age diseases, including obesity and other diseases like inflammatory bowel disease, celiac disease, etc. But there's a number of environmental contributions here that really feed into our genetic predisposition for the development of these complex disorders. And so 3 of the areas that I've been really interested in is kind of understanding how gut microbes can interact with our nutritional status or overnutrition in certain instances, as well as with our circadian systems. And while I'm not talking about sleep per se today, circadian rhythms arguably contribute to sleep patterns and sleep disruption, and how all of these factors come together to drive the development of obesity is really an important area of research, at least in my opinion. So what might be contributing to this increasing prevalence of Western disorders? Well, there is no doubt that our diets have changed dramatically, and some may consider that we have gone from more of a hunter-gatherer type population where we rely on growing or catching our own food and preparing it, to having highly processed, highly readily available, high caloric density food available to us really at all times, day or night. And these factors together have dramatically changed our eating habits, and we think that they've contributed to changing the gut microbiota. Now, this is a little bit of an older study from 2010, but I really do think it's a nice demonstration in humans on how dietary intake associates with changes in the gut microbiota. So this was a study where they actually looked at children from Burkina Faso, Africa, so a very rural, perhaps hunter-gatherer type population, as compared to a European Union cohort of children residing in Italy. And so these researchers actually looked at the 16S rRNA gene bacteria via next generation sequencing, which is a standard technique that we use now to get a fingerprint of the microbial community. And what they saw was that these two groups of children, the Burkina Faso Africa children versus the EU, had a very unique gut microbiota signature. So looking at the dominant phyla within the intestine, Firmicutes and Bacteroidetes, across these two groups of children, you can see that they are dramatically different, where the children from Burkina Faso have an expansion of Bacteroidetes, and inversely, the children from the EU, namely Italy, have an expansion of Firmicutes. And when the researchers actually looked to see what was correlating most specifically with these signatures of gut microbiota, they noted that dietary intake was one of the major drivers. And so you can see here that just based off of kcals per day between these two groups of children, there's a significant difference, as well as the macronutrient content of these diets, where the children in the EU are taking in quite a bit more fat, as well as carbohydrates. And when we think about the carbohydrates here, more than likely these carbs are simple carbohydrates rather than complex carbohydrates, which we know feeds into shaping the gut microbiota. Now, one of the major tools that we have to really understand the mechanistic role that gut microbes are playing in driving our metabolic outcomes is the use of germ-free animals, or those animals raised in the complete absence of gut microbiota. So here on the right-hand side of the screen, we have a germ-free isolator where these animals are raised and are exposed to sterile food and water. And they can live out a full, long life, even in this setting. But when we compare these animals to their conventionally raised animals here in a standard environment where we have a full milieu of gut microbiota, if we put these two groups of animals onto a high-fat diet, the germ-free animal will remain lean regardless of the diet that you expose them to, as compared to their conventionally raised counterparts that develop obesity relative to their low-fat fed counterparts. So, this highlights that gut microbes play a pretty substantial role in driving the development of disease. But then more compelling is if we take these gut microbiota from high-fat fed animals or obese animals and place them into our germ-free animals, these animals will now develop obesity and have a similar metabolic phenotype to those from their conventionally raised donors. So again, highlighting this mechanistic role. Now, there's a number of potential mechanisms for how Western diet-induced gut microbiota influence obesity, and I'm not going to go into these very, very deeply, but there's been a hypothesis that gut microbes aid in energy extraction from the diet. They can result in increased permeability that result in this low-grade inflammation in the periphery in both hepatic, adipose, hypothalamic, and even in the musculoskeletal system. There's been research to show that there's modifications of the gut-brain axis and behavioral outputs, and we've actually started to do some of this research in the context of circadian rhythms. But when we first started on this journey, we really wanted to identify novel mechanisms for the role of gut microbes in obesity. And so this was back in the day of microarray, so no fancy RNA-seq here, but we were actually comparing livers that were taken from germ-free mice as compared to their conventionally raised counterparts. And we also did a group of conventionalized or ex-germ-free mice that had received microbiota transplant from these conventionally raised animals. And we saw very similar readout as far as gene signatures in these animals. And so what was really striking to us is obviously there's a number of pathways that are differentially expressed between germ-free and conventionally raised mice, But one group of genes that kept coming up were involved in circadian rhythm signaling. And this popped up not only in the liver, but other tissues, including the intestine. And at the time, we were certainly not circadian biologists by any stretch of the imagination, but we really wanted to know, how do gut microbes contribute to, and do they have an effect on host circadian networks? Well, and why do we think that, circadian networks are so important? Well, this is a little diagram showing the major genes that are involved in the circadian gene network. And it's really made up of two limbs that contain activators and repressors that interact to both induce and suppress or degrade one another. And these interactions serve as really an underlying molecular metronome, the title of my talk, in the body that occurs over a 24-hour light-dark period and really governs our metabolic function. But there's alterations to the system that can occur, so including, genetic manipulation. So mice who genetically lack BMAL1, one of the major activators of this loop, have complete loss of circadian rhythms. But other means of disrupting the circadian clock gene network are shift work, sleep apnea, and jet lag, as well as simply shifting people to a high-fat diet. Now, to really get after how gut microbes sit and feed into the circadian network, we really need to take a multi-pronged approach. And this is really where I think my lab and our research excels, in that we are able to use a number of techniques, including in vivo animal models and in vivo cell culture systems, to really gain an understanding of the direct interaction gut microbes play with our host metabolism, as well as the underlying molecular machinery that's driving this metabolism. And our hope is really to identify a translation to humans in the clinic. And all of these components feed into one another to really aid in understanding how environmental hits, including changes in diet or sleep-wake patterns or exposure to antibodies, et cetera, drive metabolic outcomes. And so the bottom line from our early studies, and I'm not going to go too in depth on those early studies because I did want to show you more of our more recent results, is that gut microbes are really there to sense dietary cues that translate into outputs to maintain circadian networks. So gut microbes and their day versus night rhythms actually are there to sense when, how much, and what we're eating. So in the case of a low-fat, high-fiber diet, we see that there's microbial oscillations in the community as far as who's there during the day versus the night. And this gets translated into microbial signals, what we call the metabolome, that then can be absorbed by the host and can influence circadian networks within various tissues, including the central clock located within our brain, as well as peripheral clocks that are located in peripheral tissues like the liver. And when all of these things are aligned, microbial oscillations and our circadian networks are in alignment, and this promotes metabolic health. And so microbial signals that we think are interesting are short-chain fatty acids, which are derived from the fermentation of complex carbohydrates or a high-fiber diet. But we know that there can be certain hits to the system, like high-fat diet, for instance. And so when you take in a high-fat diet, you decrease the microbial oscillations. So who's there changes and their rhythms are actually dampened, which then disrupts their microbial signals, or the metabolome becomes disrupted, or there's unique metabolites that are made that can then influence the host circadian networks differentially. And this can actually result in misalignment of these various portions of the network and result in the development of obesity. But we know that this is a bidirectional communication that occurs. So we know that if we disrupt host circadian systems, this can actually lead to gut dysbiosis, which then can feed back to altered metabolism. So for example, if we impact circadian networks by either shift work or sleep disruption, or we can actually knock out genes, as I mentioned, like BMAL1, and we can see that when this occurs, that the oscillations of the gut microbiota actually change and become disrupted as well, disrupting the microbial metabolome again, which then can feed back and impact host circadian networks negatively, resulting in metabolic dysmetabolism. So as I mentioned, genetic manipulation or jet lag on the host side can disrupt microbial oscillations, and this can also result in obesity. But we know that we can actually manipulate microbes to restore host circadian dynamics of the host to alleviate obesity as well. So even in the case of high-fat diet, where we have disrupted microbial oscillations of both who's there and the functional outputs that they're making that are then absorbed by the host, some beautiful work by Amir Zarinpar and Sachin Panda showed that even if you time restrict high-fat diet, so if you time restrict feeding to the dark cycle for mice, for example, because they are nocturnal, so this would be a time when they would typically be eating. So if you restrict this high-fat diet to the dark cycle, you can actually restore some of the microbial oscillations and the functional outputs. And this restores and realigns circadian networks and can prevent the development of obesity. Now, there is some work in humans to show that early versus late eating can impact diurnal patterns of salivary microbiota in healthy humans. And so, as you can imagine, in mice, we have the luxury of sampling stool or collecting regional samples over a 24-hour period. But this becomes remarkably challenging in humans, as you can imagine. And so what Collado et al. Was able to do was actually sample saliva over a 24-hour period. And then they also sampled, they had these humans on either a late eating, so later in the day, they were taking in the majority of their diet, or they were taking in an early diet. And then they also did a washout period and a crossover design. And they showed that the oscillations of the gut microbiota were inverted in the salivary microbiota, depending on whether the person was eating late or early. So this is some evidence, anyways, direct evidence, that we can detect microbial oscillations in humans and that they are indeed tied to our time of eating. Now, more recently, the same researchers performed another study where they were actually looking at the time of intake for a daily bolus of chocolate, which I would like to sign up for this study. But what they did was they had menopausal women, 19 of them, who were given a bolus, 100 grams of milk chocolate for 28 days was the intervention. And they either had no chocolate, a morning chocolate, or an evening chocolate. So again, this was a crossover design with a washout period in between. And while they didn't actually examine the oscillations of the gut microbiota in this condition, what they did was they measured various parameters. So both who's there in the gut microbiota, as well as some of the outputs that they make, like short chain fatty acids. And what they noted was that regardless of whether or not the people or the women were taking in chocolate, either evening chocolate or morning chocolate, so EC or MC, there was an increase in certain short chain fatty acids relative to those humans who were not taking in any chocolate. So this suggests that perhaps fiber component or other components within chocolate could actually be promoting fermentation of specific microbes within the community. And so when they actually looked at the short chain fatty acid profiles as compared to the 16S profiles of the types of microbes that were there, they noted that there was a very unique signature. So this is a principal coordinate plot. I always say that this is a little bit like Sesame Street. So one of these things is not like the other, and how can we bin them, you know, to show similarities or differences. And so ultimately you can see that there's a very nice separation here between individuals who are taking in no chocolate, morning chocolate bolus, or an evening chocolate bolus that related to both who was there in the microbiota community as well as the short chain fatty acids that were produced. And so if we take this principal coordinate plot with the different colors for no chocolate, morning chocolate, or evening chocolate, and we look at the percent of certain microbes like Ruminococcus or Lactobacillus within the microbiota community relative to acetate, for example, you can see that there's a positive correlation here between the percent of Ruminococcus within that community for both evening and morning chocolate consumption relative to no chocolate for the amount of acetate that's produced. And then the flip side is true, although not as stark, for Lactobacillus, where there's an inverse relationship here with chocolate intake versus no intake for the percent of Lactobacillus and acetate that's being produced. So what this group showed was that there were really no overt changes in weight within these women who were taking in chocolate, which was a bit surprising because this was, a pretty high caloric bolus of food that was taken in every day by these people. But what they did notice was that morning chocolate actually enhanced lipid oxidation and decreased fasting blood glucose, whereas evening chocolate consumption actually increased skin heat dissipation and promoted regular sleep episodes. So, depending upon the person's chronotype and the time of delivery, you can actually alter their metabolic outputs in concert with associations in changes in the gut microbiota. But even though this is really nice evidence to support this idea and concept that, food intake by mouth is required to drive gut microbial oscillations and community membership, we actually wanted to test this. So how important is food intake to drive microbial oscillations? And so with a group up here at UW-Madison, before I was even here as faculty, we collaborated with a group out of the hospital who actually could do enteral feeding or parenteral feeding, and not in humans, but in mice, which I think is actually even a bit more challenging. And so this was actually performed with another researcher who I think you heard from earlier this year, Joseph Pierre. And so we actually looked at gut microbiota over a 24-hour period in enterally fed versus parenterally fed animals, where parenterally fed animals are infused with nutrients into the periphery continuously over a 24-hour period, whereas their enterally fed controls were actually able to eat by mouth, and they were eating a low-fat, high-fiber diet. And we were able to look at the microbial oscillations here. And surprisingly, we noted that there were still microbial oscillations that were evident in the community in the parenterally fed animals. So here you can see that 3 of the major phyla in the intestine, Bacteroidetes, Firmicutes, and Verrucomicrobia, exhibit oscillations or a day versus night pattern here over the 24-hour light-dark cycle in the context of relative abundance in the enterally fed animals. And while we see a very different community membership here with this expansion of Verrucomicrobia, we still see that there is a diurnal pattern to that gut microbiota over the 24-hour light-dark cycle, even in our parenterally fed animals, suggesting that there are other factors than just food intake by mouth that can drive diurnal oscillations in the gut microbiota. So we really wanted to know, how is that possible, and who and what is contributing then, if it's not just food intake by mouth, to driving diurnal oscillations in the gut microbiota? And so our very astute colleague, Joe Pierre, was actually really interested in understanding how, the changes in the innate immune system in the gut following parenteral feeding. And so he was interested in antimicrobial peptides specifically. And he was looking at one in particular called regenerating islate-derived protein 3 gamma, or Reg3 gamma. And so Reg3 gamma is a really interesting antimicrobial peptide that's made predominantly in the small and distal small intestine of humans and animals. And this is a TLR and MyD88 dependent antimicrobial peptide. So what happens is there's various microbes within the community that bind to TLRs and induce MyD88 signaling, and ultimately through a number of steps that aren't shown here result in release of Reg3 gamma within the luminal space, and this can actually result in killing of certain microbial community members. So for example, Reg3 targets gram-positive organisms, so organisms like Listeria monocytogenes, which many of you know is a foodborne pathogen. And so Reg3 gamma is one of the essential components for killing foodborne pathogens like Listeria following its release. So this aids in maintaining microbial homeostasis in the intestine and can prevent infection. So what Joe noticed was that when we parenterally fed these animals, he was looking at Reg3 gamma expression in the distal small intestine, that our enterally fed animals who were given regular chow over a 24-hour period actually exhibited a nice diurnal pattern in Reg3 gamma expression over a 12-12 light dark cycle. But our parenterally fed animals actually lost oscillation in Reg3 gamma. And we thought this was very intriguing. And right about the time that we made this initial discovery, another paper came out in 2014 to show that if you antibiotic deplete the gut microbiota, so AIMD, which is antibiotic-induced depletion of the gut microbes, and you look at Reg3 gamma expression relative to the untreated controls, you can see that there is also a loss in diurnal rhythms of Reg3 gamma expression as compared to the control counterparts. So this was very intriguing to us because now we had two ways of creating what we consider to be a gut dysbiosis that were leading to the same outcome from a circadian perspective. And we thought, well, maybe it's really not the circadian system and circadian network that's in control of something like Reg3 gamma, but it's actually a communication between the gut microbes and the host immune system. So this really led us to want to understand what are the dominant versus secondary drivers for microbial oscillations. So clearly we have a core or central clock within our brain that dictates the majority of our circadian networks and maintains an alignment. But we also have time feeding, and we know that that's important as well for driving gut microbiota oscillations. And then now we have this understanding that perhaps the innate immune system or immune parameters within the gut may actually communicate with the host circadian clock to drive diurnal rhythms. And so this is, and what are the bidirectional communication factors that are occurring here? And we decided to kind of drill down on this idea of the gut microbes driving diurnal rhythms of specific host immune parameters that aid in maintaining this circadian dialogue between the gut microbiota, our dietary intake, and host immune function in the context of metabolism. So in order to do this, we went back to our germ-free versus conventionally raised counterparts here, and we fed them either a regular chow or a high-fat diet. And our first go at this was to look to see how diet-induced gut microbes, or the lack of them, could actually impact diurnal Reg3 gamma in the context of circadian gene expression patterns as well. And so what we showed was that really when we looked at BMAL1 and PER2, which are two key genes associated with our circadian network, we noticed that really there was no difference between groups, regardless of diet or presence or absence of gut microbes, in the diurnal patterns of both BMAL1 and PER2 expression over a 12-12 light-dark cycle here. But on the flip side of this, we noticed again that antimicrobial peptides, and specifically Reg3 gamma, exhibited a diurnal pattern over this 12-12 light dark cycle, really only when our conventionally raised animals here were fed regular chow diet. Now, if these animals were placed on high-fat diet, we lost this diurnal expression of Reg3 gamma, and this was nearly absent in our germ-free component, in our germ-free counterparts, indicating that both gut microbes and diet can actually shape expression patterns of an antimicrobial peptide like Reg3. But when we looked at other antimicrobial peptides like lysozyme, we found that there was really no apparent pattern that was related to either diet or presence or absence of gut microbes So we began to then really have evidence to drill down further on Reg3 gamma. So in our SPF, or conventionally raised counterparts, we actually then were able to look at the diurnal oscillations within the small intestine to see how diet was actually shaping both community membership and what they were doing during the day versus night. And I will say that we backed up just a little bit to confirm that our expression patterns by mRNA actually were observed at the translational level as well. So this is just immunostaining at 3 different time points during the day. So these 2 time points, Zeitgeber 2 and 10, are during the light cycle, and ZT18 is during the dark cycle. And you can really see that there's evidence of diurnal expression of Reg3 within the epithelium, only at ZT10 in the regular chow-fed animals. And while there is some expression in high fat, it's not nearly as evident from a circadian perspective as it is in regular chow-fed animals. And here's the quantification of our immunostaining here for our regular chow-fed animals versus high fat. And then really, when we compared this to lysozyme, there was no evidence of diurnal expression and no difference between diet. And then we also confirmed this by Western blot as well. So regular chow-fed animals had higher expression beginning at ZT10, whereas in high-fat fed mice, Reg3 gamma expression remained low relative to housekeeper gene. And then finally, we were interested in kind of understanding where in the epithelial compartment are we losing diurnal expression? So, Reg3 gamma can be made by several different cell types in the intestine, including Paneth cells, which sit at the base of the crypt of the gut. But they can also be made by absorptive enterocytes as well, and other cells along the crypt villi axis. And so this is where we wanted to actually look to see where and who is losing oscillation on the host side for Reg3 expression. And we showed that really it doesn't have much to do with the Paneth cell at all. So when we actually fractionated the crypt villi axis, there was no evidence of diurnal differences in the crypt, but we saw that the epithelial component, or the absorptive epithelial component, exhibited a diet-dependent expression, as well as a time-dependent expression. So high-fat fed animals exhibited low expression regardless of time of day, whereas there was this diurnal or possible diurnal expression pattern observed only in regular chow-fed animals. So then next we wanted to look to see what was going on with the diet and the gut microbiota. And so here we actually were able to take the distal ileal contents from our conventionally raised regular chow versus high-fat fed animals, and harvest the distal ileum over a 24-hour period and perform 16S sequencing here. And so this is a principal coordinate plot, again, showing how different the high-fat fed community is relative to the regular chow-fed community. And when we look broadly at the phylum level changes, we saw that high-fat diet resulted in an expansion of firmicutes and a reduction of bacteroidetes, which has been shown by other groups as well. But really, we were interested in the oscillatory capacity here. And so, we looked at the percent oscillating versus non-oscillating OTUs over this 12-12 light dark period. And so in regular chow animals, we could see that, about 20 to 30% of the community was exhibiting oscillations, whereas in their high-fat fed counterparts, there was a decrease in the percent of the community that was oscillating. So we wanted to know whether or not specific taxa could correlate with Reg3 expression. And so certainly, indeed, we noted that there was a positive correlator. So when we looked at Reg3 gamma expression here in our regular chow in blue, high fat in red, relative to the relative abundance of this specific lactobacillus operational taxonomic unit, we could see that there was a significant positive correlation. So as Reg3 expression was increasing over time, there was an increase in this specific lactobacillus within the community. But on the flip side of that, there were negative correlators. So again, with Reg3 gamma expression being extremely low in the high-fat fed animals relative to these other, these two Clostridiaceae and Peptistreptocaceae family members, we could see that there was a strong and significant negative correlation. So if these community members were actually increased in the community due to high-fat diet, Reg3 gamma expression was low. And this was true when we actually looked at the relative abundance of these OTUs over time. So this lactobacillus obviously has increased relative abundance as compared to high-fat fed animals. And our negative correlators actually expand in our high-fat fed animals and are at very low relative abundance or are non-existent over the 24-hour 12-12 light dark cycle. So our interim conclusion here is that Reg3 gamma exhibits clock-independent diurnal expression patterns in the distal ileum that are driven instead by the presence of diet-induced gut microbes. And feeding a high-fat diet actually shifts the distal ileum bacterial community membership and reduces the percent that exhibits diurnal oscillations as compared to regular chow-fed animals. We think that high-fat diet alters the oscillations of specific taxa within this community, and these diet-dependent taxa actually correlate with diurnal Reg3 gamma expression patterns. So our next adventure was to actually see if specific gut microbiota communities or bacterial strains selected by diet can influence Reg3 gamma expression. And so our first go at this was to prepare the distal ileal content lysate from these differentially fed animals and expose them to enteroids or organoids in vitro setting, or we prepared conditioned media from our positive correlators and negative correlators and exposed organoids to these. So organoids, for those who are not familiar, are actually stem cells harvested from the ileal crypt base. They can be harvested from throughout the intestine, but we chose to harvest them from the ileal crypt base of wild type animals. Nearly all the cell types that you would see in the intestine are distributed and recapitulated in these organoids, and many of the functional characteristics are maintained, and we actually call these mini guts. So when we actually exposed our organoids to the ileal content lysate from regular chow-fed animals, as predicted, we saw an increased percent induction of Reg3 expression relative to the high-fat fed ileal content lysate. And then when we looked actually at our positive correlator, Lactobacillus rhamnosus GG, as compared to our negative correlators or Peptistreptocaceae and Clostridiaceae strains, conditioned media, we saw that really only LGG was able to induce Reg3 gamma expression relative to our negative correlators. So suggesting that specific diet-induced gut microbes can contribute to Reg3 gamma expression. And we did notice that there was MyD88 dependency to this. So when we compared LGG to P. stomatis in MyD88 heterozygote versus knockout enteroids, we saw that only those that were MyD88 heterozygote had the capacity to upregulate Reg3 gamma expression, and this was only in the case of LGG. So P. stomatis was not able to induce as expected. And we also noted that the expression of Reg3 was driven, we think, by small molecules. So this was conditioned media that was fractionated just by size. And we could see that LGG was able to induce Reg3 gamma regardless of size. And so even that fraction that was lower than 3 kD was able to induce to an equal degree. And there was actually no induction as expected with P. stomatis conditioned media fractionation. And then this is a more recent piece of data where we actually heat treated the conditioned media from the 3 kD LGG, and we noted that there was a significant reduction in its ability to induce Reg3 gamma expression relative to the unheat treated 3 kD LGG. So we think that diet-induced microbial communities can differentially influence Reg3 gamma expression in vitro, and that individual bacterial reference strains of those selected by diet can actually directly induce or suppress expression both in vitro and in vivo, and I'll show you the in vivo data next. So L. rhamnosus GG could actually induce expression, whereas Clostridiaceae and Peptistreptocaceae conditioned media are indicators of negative correlators with Reg3 actually inhibit Reg3 gamma expression. And we think that this is small molecules made by specific microbes that can induce or suppress Reg3. So we then wanted to take this back in vivo. So we monoassociated germ-free mice with L. rhamnosus GG to see what their capacity was to induce Reg3 gamma and whether or not this was in a diet-dependent manner. So we had our germ-free mice that were either fed regular chow or high-fat diet, and they were monoassociated with Lactobacillus rhamnosus GG. We did not have the fluorescent version of this. This is a picture I stole from somebody else. But importantly, we took these animals off at 2 times of day, ZT2 and ZT10, which is the predicted time when Reg3 gamma should be at its highest expression. And we noted that, as expected, germ-free mice didn't exhibit a dramatic increase in Reg3 gamma expression regardless of diet or time. But really, regular chow-fed animals had an elevated level of expression as compared to both their germ-free and high-fat fed counterparts. And there did seem to be some evidence, although not significant, of time dependency to this expression. So our model here is that we think that low-fat diet and diet in general is serving as the primary driver and driving a small intestinal eubiosis, and is a primary driver of microbial oscillations. And so this leads to an expansion and diurnal variation in healthy community members, like Lactobacilliaceae community members, that can then upregulate Reg3 gamma expression as well as diurnal patterns of its expression. And this upregulation inhibits other members of the community from expanding as well as from oscillating. So for example, Peptistreptocacaceae then is inhibited within this community. And this results in leanness. But on the flip side, when you take in a high-fat diet, you disrupt the small intestinal eubiosis and create a dysbiosis. This results in a decrease in those oscillators and perhaps a gain of other oscillators that you don't want to be oscillating. This results in a loss of functional members of the community like lactobacillus, no signals for driving Reg3 gamma oscillations, and then this allows for this expansion of members of the community induced by high-fat diet like Peptistreptocacaceae. So key takeaways here are that time of feeding coupled with diet composition are really primary drivers of microbial community membership and the broader oscillatory capacity. But there's key host innate immune components like Reg3 that oscillate independently from the core circadian clock. And the diurnal Reg3 gamma expression actually depends on the presence of select community members. And this bidirectional communication between Reg3 gamma and gut microbes serves as a secondary driver to maintain local intestinal oscillations, which may, in the long term, affect metabolic homeostasis. So when we think about this in the context of future of the chronobiome work, if you will, another biome, the gut microbiome is a key feature of maintaining circadian networks. I think that's really apparent, and it has to be considered as a influencer of host circadian rhythms. Whether or not this could be impacted by time delivery of either prebiotics, probiotics, or even postbiotics, so things like short chain fatty acids, could impact their influence on promoting restoration of the gut microbiome and host health, requires exploration. And I think in order to do this, we need to have deeply phenotyped subjects. So we need to understand their sleep-wake patterns, feeding patterns, work schedules. So really their chronotype is essential. So diurnal patterns of microbes in deeply phenotyped subjects are already being used to predict metabolic disease outcomes. And really, I think that this is evident that there's a no one-size-fits-all approach. We really need personalized medicine and personalized nutrition here. And the key is understanding unique chronotypes. So for example, are you an owl or a lark? And with that, I just wanted to acknowledge my collaborators and as well as my funding sources. So as I mentioned, a lot of this work was done at University of Chicago with my former mentor, Eugene B. Chang, and a very talented graduate student, Katya Frazier, who will soon be on the market looking for a postdoc soon, and Amal Kambal, who was a technician in our laboratory, and then as well as Dr. Joseph Pierre, who is coming back to UW-Madison here very soon. And then as well as my funding strings, as was mentioned earlier, I do have a K01 Career Development Award that funded the majority of this work.